So it's my great pleasure to get to introduce Surya Ganguli, who is from Stanford by way of Berkeley. And Surya and I overlapped at Stanford. He arrived to great fanfare sort of maybe a couple of years before I left as a postdoc. And I have these very good memories of being in Pete's Cafe with Surya, arguing over some problem or another that was lost to time. But he was always sort of very patient with me and very creative, and we always had these very interesting discussions. And so I feel like I knew Surya quite well, and that sort of continued sort of through the years since. And, I'm aware of some of the nice awards that he has. He has, for example, a Simons Investigator Award, a McKnight Award. But then there's this interesting thing that happens when you're asked to give an introduction, which is you get to download somebody's CV, and it feels a little sneaky, right? You sort of like you're spying on them. And there's a number of things I'd like to share that I won't in the interest of time, but the one thing I will, just mostly in the interest of embarrassing Surya, is that he graduated first in his class in high school at the age of 16 and has, somewhat disturbingly, 3 different bachelor's degrees and 3 different master's degrees. And so with that, I will call Surya out to
Thanks. It's great to be at Columbia. It's great to see Mark again. I do remember very fond memories of discussing science with you at Pete's at Stanford. I also, like almost everybody else at this conference, was trained at Columbia. I spent a year here, and I will forever have fond memories of my time here. And it's wonderful to see such an amazing community being built up, to see this new building. I wasn't in this new building, when I was here. My first paper was actually in neuroscience, was with Ken Miller and Mike Shadlin and Mickey Goldberg, who are all here. So I wanted to talk about self-organizing neural representations of space through exploration. I wanted to start by thanking my collaborators. This is a series of papers that I've done in close collaboration with Licia Giacomo, without whom none of this would be possible, and a very talented group of grad students and postdocs. Right? So if we think about our modern conception of space, it's absolutely astounding. Right? Less than 100 years ago, Einstein came up with the equations of general relativity, which can be summarized as matter tells space and time how to curve, and curved space and time tells matter how to move. This is an astounding theory, and in less than 100 years, we had direct experimental tests of new concepts that arose from this theory. Gravitational waves as ripples in space and time itself, and black holes as occurring when space and time curve so much it generates a singularity. It is far beyond the ken of modern neuroscience to understand how the human mind could conceptualize space and time in such a way that is so far beyond the reach of ordinary experience. However, the problem of reasoning about space is an ancient problem that early animals had to solve. And I'll speculate wildly about how solving that ancient problem might eventually lead to this at the end of my talk, but let's talk about that ancient problem. Let's say that an animal, or equivalently a robot, because this is a pressing problem in robotics right now, an animal or a robot enters a new environment it's never encountered for the first time. Say this poor Roomba, all right? It's got to figure out where it is. It's got to localize itself. But how the heck does it localize itself in a new environment when it doesn't even have a map of the environment? So that's the localization problem, okay? So then the next step is it's got to build up a map of the environment. But how does it build up a map of an environment if it doesn't even know where it is in the environment? So there's a simultaneous nested chicken and egg problem, which is called simultaneous localization and mapping, or SLAM in the robotics literature. And what we're going to explore is we're going to explore how known mechanisms of local synaptic plasticity and neural dynamics can conspire to learn a map of a new environment over time through exploration. Right? So, of course, deep within the brain, for example, in the entorhinal cortex, far from the sensory-motor periphery, the brain has created very, very striking maps of space. For example, the famous grid cells, landmark cells like border cells, speed cells whose firing rate increases monotonically with speed, head direction cells whose firing rate depends on the direction of your angular direction of your head. Okay, but it's important to note that these high-level correlates of space don't appear for free. Right? Instead, you have very, very low-level streams of photoreceptor activations proprioceptive sensors, efferent motor commands. And from this stream of ongoing sensory motor experience through a sequence of computations, you have to construct an internal estimate of position. I'm going to focus on the last stages of this computation. How we know where we are can be derived in two distinct ways. We could integrate our steps or our velocity to compute position, but we could also correct our internal estimate of position through landmarks. And I want to study how this works. Okay? So a fundamental consistency requirement governing any spatial map-forming circuit is the following consistency requirement that relates path integration to landmarks. Let's say, for example, an animal encounters a landmark. Sensory perception of that landmark triggers a neural activity pattern. Okay? And then path integration may move that activity pattern as the mouse moves along, generating a new pattern B prime. Alternatively, the animal might directly encounter this landmark, and sensory perception of this landmark will generate an activity pattern B. Consistency demands that the activity pattern elicited by sensory perception of this landmark is equal to the activity pattern generated by taking this path. Otherwise, internal representations of the circuit will not be in one-to-one correspondence with space, independent of where you came from. All right? So how does plasticity and neural dynamics solve this consistency problem? And can we use it, for example, to model grid cells? Okay, so the very first thing that Lisa and I did together was we first assessed to what extent landmarks did indeed correct the grid cell representation. So these grid cells are stable over long periods of time, at least for the hour over which you record. Where does the origin of this long timescale stability arise from? Is it a very precise path integration mechanism, or is it continuously getting corrected by landmarks? And it turns out it's the latter. We showed that the errors in the spike, we devised analyses to show that the errors in the internal estimate of position grow strikingly rapidly, almost doubling within a timescale of 40 seconds. So were it not for the fact that the mouse is encountering borders almost at a rate of 2 to 3 times a minute at least, you would never ever see grid cells by averaging over an hour. So path integration is incredibly imprecise in the circuit, and landmarks are continuously correcting the internal estimate of path integration here. Okay? So now with that evidence in mind, we generated an analytically solvable attractor model where we had plastic synaptic inputs from landmark cells, and we proved that it could learn a consistent spatial map. The model made predictions, particular history dependence in grid cell firing fields, and it explained why grid cells get deformed in irregular boxes. And then to probe the model and the brain further, we devised virtual reality experiments where we altered the very laws of physics themselves. In particular, we altered the gain between how many steps you take and what you see in a virtual world. So we kind of break the system, and we find two dynamical regimes. One where if landmarks and path integration disagree a little bit, the grid cells will phase shift. On the other hand, if they disagree a lot, the grid cells will break free of the landmark reference frame and completely remap. And we see both of those in data. So that's kind of where we're going in this talk. Okay, so what is the model that we came up with? We combined a lot of technology, old technology in theoretical neuroscience, and then added some new ingredients, which we'll get to. So one piece of old technology is the ring attractor network. So we're imagining that this is, you can think of this as a 1D model of grid cells. Imagine a set of neurons that for pedagogical purposes we just arrange in a ring, though in the brain it doesn't need to be that way. But we imagine the connectivity is such that you have local excitation and long-range inhibition and appropriate nonlinearities, such that this ring attractor is capable of sustaining a stable bump of activity, and that because the connectivity is translation invariant, that bump could appear anywhere on the ring. Okay? All right, now in order to path integrate, we need this bump. Oh, so you can summarize the activity of this entire network by what we call the attractor phase, which is the phase on the ring at which the bump attains its peak activity pattern. Okay? So we'll call this the attractor phase. Now what we would like is to do path integration, we'd like velocity to influence the motion of this bump. Okay? So the way we do that is we use an old idea from Sansanovich and McNaughton that was reproduced in Barak and Feet, in their model of grid cells. The idea is we have velocity-sensitive cells, which are also found in medial entorhinal cortex, that have conjunctive position velocity dependence. So for example, if the mouse is moving east, this cell will fire, and its connections onto the attractor network are slightly shifted. And the shift will cause the bump to move clockwise. Alternatively, if the mouse is moving the opposite direction, there's another population of cells that will have a shifted inputs here that will cause the bump to move counterclockwise. And you can set it up so that the rate of change or rate of phase advance of the attractor phase is linearly proportional to velocity so that this thing will path integrate. So the phase as a function of time will be the integral of velocity. Okay? So this is actually a 1D model of a grid cell. Why? Imagine that you're an electrophysiologist eavesdropping on one neuron in this attractor network. As the mouse is going from, say, west to east, the bump will go round and round, and this grid cell will firing will go up and down periodically. And all of these other cells are grid cells with the same period, but a different phase. And you find such grid modules at different dorsal-ventral levels of the medial entorhinal cortex. Okay, so now the new ingredient is, so as of yet, there's absolutely nothing that anchors the bump to an absolute position in physical space. It just gets pushed around by velocity. There's no way to correct the path integration mechanism. So the new ingredient that we add is landmark cells. So these are cells that fire if and only if the mouse is in a certain region of space. A prototypical example would be a medial entorhinal border cell, for example. We imagine that this landmark cell provides excess excitation to some point, any point, on the attractor ring. So what would its effect be when the mouse encounters a landmark? It'll cause the attractor phase to become attracted to the position of peak excitation of the synaptic weight profile. So we'll call the position of this peak synaptic excitation the pinning phase of the landmark cell, because when the landmark cell fires, it pins the attractor phase of the attractor network to the pinning phase of the landmark cell. Okay? All right, now immediately we run into the problem of self-consistency. You can see it in pictures here. Imagine, for example, the mouse is moving around, and the path integration happens to move the attractor phase to this location on the attractor network when the mouse encounters a landmark associated with this landmark cell. In a new environment, there's absolutely no guarantee that the phase that the attractor network arrives at due to path integration equals the pinning phase of the landmark cell when it arrives at the landmark. Okay? So we need a new ingredient to enforce that self-consistency, and that ingredient will be Hebbian learning of these afferent synapses from the landmark cell onto the attractor network with some weight decay. So imagine, for example, the bump is here. These cells are active, and so is the landmark cell, because it's at the landmark. And so Hebbian learning will strengthen these synapses, and weight decay will weaken these synapses. And as you can see, Hebbian plasticity, at least in a cartoon-like fashion, could actually solve this self-consistency condition. Of course, it's a much more complicated system because you have this coupled dynamics between neurons and synapses where the pinning phase of the landmarks are dragging around the attractor phase, and the attractor phase is modifying the pinning phase of the landmark cells. Now, I've been explaining everything in cartoons, but don't worry, version number 1, there are actually equations underlying all this. So although it may seem like I'm totally blowing smoke, I'm not. Don't laugh too hard. But don't worry, version number 2, I won't show these equations again. Okay, so everything's mathematically well-defined. We can even prove theorems about this, simulate this, and all that. But I can explain everything in pictures, and the pictures do reflect the equations. Okay, so now how is it? Does this cartoon-like picture solve the self-consistency condition? The way to see how it might work is to work out the model in the simplest possible scenario, where a mouse has entered a new 1D environment where there's two borders, a west border and an east border, and it doesn't know the length of the environment at all because it's never been in this environment before. So in some sense, how does the mouse learn about the length of this environment? So let's simulate the model in our heads. So initially, it starts off at the west border. The west border will have some pinning phase. Okay? The attractor phase will be pinned to the west border cell's pinning phase. Then path integration will advance the attractor phase to some new location, but because this is a new environment, there's no guarantee that the east border cell's pinning phase equals the phase arrived at by the attractor network, or the grid cell network. However, Hebbian plasticity will move the pinning phase a little bit closer to where the attractor phase ended up, and then, of course, the pinning phase will pin the attractor phase. Okay? Then path integration will occur in the reverse direction until you end up here. Again, there's no consistency to begin with, but Hebbian plasticity will move the pinning phase slightly over, And of course, then it'll pin the attractor network. And then, over a few cycles of exploration, depending on the time scale of plasticity, you'll end up with this happy equilibrium configuration of the neurons and synapses, such that the pinning phase associated with the west border cell plus the phase advance due to path integration arrives exactly at the pinning phase of the east border cell, and vice versa. And in this happy situation, the phase assigned by the attractor network to any interior point in physical space is independent of where the mouse came from, at least in one dimension. Okay? So now this circuit does indeed map space in a one-to-one fashion in the sense that individual spatial positions are assigned to grid cell firing patterns in a one-to-one fashion, and there's a consistency between landmarks and path integration that's been learned over time through exploration. Okay, so this could be viewed as a simple neural solution to the SLAM problem. Okay? To make contact with experiments, we can generalize everything to 2 dimensions. It's conceptually identical. The ring of neurons gets replaced by a sheet with local excitation, long-range inhibition. Stable patterns now become hexagonal firing patterns on the sheet. The attractor phase gets replaced with a point in the torus. And a border cell becomes a particular pattern of excitation in the torus that can be viewed as a line along this torus. If not all of this made sense, don't worry about it. You don't need to understand this in detail. But everything that conceptually worked in 1 dimension also works in 2 dimensions, or any arbitrary number of dimensions. Okay, so now we actually have a general mathematical proof that the coupled dynamics of neurons and synapses does indeed solve the self-consistency condition that relates landmarks to path integration in the following sense. An English language version of the theorem is that for any number of localized landmarks, you could have 10, 15, 20 landmarks in some very landmark-rich environment, in any number of dimensions, 1, 2, 3, even higher if you want, learning self-organizes all of the landmark cell pinning phases so that for the pinning phase of any landmark A, plus the phase advance due to path integration and going from landmark A to landmark B, exactly equals the pinning phase of any other landmark B. Okay? And you can find the proof in this paper along with experimental predictions. Okay? So now let's go through some predictions. Okay? So in 2 dimensions, where you might have border cells along every single wall, so you have 4 border cells to deal with, things become a little bit more complicated. But you can summarize a prediction of the model as the following. In 2 dimensions, there should be a small history dependence in the grid cells, because the border cells are always correcting the grid cells, and it could go through multiple pathways between pairs of borders. It turns out that on average, the grid cells will be sucked in a little bit towards the last border the mouse came from. That's a prediction of the theory. Okay? So for example, if you're coming from the east border, the grid cells will be, if you average over all trajectories coming from the east border, the grid cells will be sucked a little bit towards the east. If you come from all trajectories from the west border, they'll be sucked a little bit towards the west. Now, in real data, grid cells are noisy. You have finite numbers of spikes. How do you detect this? You can cross-correlate with a spatial lag all of the west-conditioned trajectories or firing fields of all the grid cells and spatially lag them and ask how much do I have to shift the west-conditioned firing fields to map the east-conditioned firing fields. Okay? And the prediction is for reasonable values of model parameters, you have to shift the west-conditioned firing fields about 2 centimeters or so east to match the east-conditioned firing fields. And similarly, for the south-conditioned firing fields, you have to shift them 2 centimeters north to best match the north-conditioned firing fields. And that's exactly what we see in the neural data. This is a peak in the cross-correlogram between the west-conditioned firing fields and the east. And you see you have to shift the west-conditioned firing fields a small number of centimeters east to match the east-conditioned ones, and you have to shift the south-conditioned ones about 2 centimeters north to match the north-conditioned ones. Now, this small history dependence need not be a bug. It could be a feature. Whenever you're trying to estimate a noisy quantity, like for example, by integrating noisy velocity, if you have some prior information, you can often come up with a better estimate by incorporating your prior to obtain a biased estimator as opposed to an unbiased estimator. So this slight bias in your internal estimate of position towards the most recently encountered border might be actually an optimal solution in the face of noise and path integration. And we're looking into Bayesian models of that now. Okay. Another famous result that was previously unexplained is that if you put mice into an irregular container, the grid cells will curve in a certain characteristic way. Okay? So the grid cells, they model this as individual firing fields behaving like van der Waals particles that locally attract and long-range repulse each other, so it looks like a piece of jello deforming, but that can't be a biologically plausible model. But it turns out that this actually arises for free in our model if you have border cells here. The basic intuition is the border cells will pin the attractor phase along a certain curve on the torus. And so the next time the grid cell can fire is when the phase of the attractor network moves away from the pinning phase of the border cell. And that's why the grid cells become sensitive to the angular orientation of the borders. Okay? So that's the basic idea. Okay, so now let's probe this system further. And really, a long history in neuroscience is to probe a neural system or a biological system, break it, and see how it responds to breakages. So the way that we're going to try to break the system is, again, we're going to alter the laws of physics. We're going to alter the gain between how much an animal moves and what the animal sees in a virtual environment. And so let's show you that. So this is a classic, and these are all experiments done by Malcolm Campbell and Lisa's lab. This is a classic experiment of a mouse, a head-fixed mouse on a trackball. And I don't think the video, let's see, the video does not work. That's fine. So what happens is the mouse moves on the trackball, and this cue-rich environment, this 1D track, advances in the visual field of the mouse in proportion to how fast the mouse is moving. So the mouse has many encounters with this environment. It gets used to it. Okay? Then we find that grid cells become grid cells, and grid cells in the 2D world become grid cells in this environment. Border cells in the 2D world become landmark tied cells in the 1D environment, and so forth. Then what we do is we change the gain between number of steps taken on the ball and the rate of advance of the environment. Okay? So you could do either a gain decrease, where for each step the environment comes more slowly, or you could do a gain increase, where for each step the environment comes faster. Right? And the question is, how will the grid cells respond to this altered virtual reality with the gain change? Right? So as usual, the model came after the data, but the data was very hard to understand. And so for a brief period of time, we actually ignored the data and tried to imagine what our model would say. And that actually taught us how to analyze the data and understand it. So how would you model this altered virtual reality environment? It turns out you can model the entire thing using just two coupled oscillators as follows. These are not real oscillators in the brain, but effective phenomenological oscillators. Okay? So the first oscillator is, as the mouse is moving on the ball, you can imagine that proprioceptive input from the motor system is giving you a velocity signal, a motor-driven velocity signal that's driving path integration in an attractor network. So as the mouse is moving, the attractor phase is driven by path integration at some frequency. And that frequency is the gain between velocity measured by leg motion or trackball motion, and that drives this attractor. So that's one oscillator. Okay? But there's a second kind of implicit oscillator. As these landmarks are coming at the mouse, each landmark will have learned a pinning phase. So for example, the first landmark will become active. It'll give you a pinning phase here. A second landmark will become active, a pinning phase here, and so on. So now you have a visually driven oscillator that's a sequence of pinning phases. Okay? If the mouse has encountered this environment for lots of times, i.e. the baseline condition, it'll learn a situation where the pinning phase of each landmark equals the phase that path integration driven by motor inputs gets you to. Okay, so everything's consistent. That means the two oscillators have the same frequency, they synchronize, and they both move along together. Okay? You can model the gain change experiment by changing the frequency of, say, the landmark-driven oscillator. Okay? Because the landmarks are arriving, say, at a higher rate or a lower rate. So now you'd have two, in the gain change experiments, you now have two coupled oscillators at different frequencies. And we know a lot about such systems. If the frequency difference is small, i.e. landmarks and path integration don't disagree too much, okay? So yeah, this is baseline, where landmarks and path integration agree. If landmarks and path integration don't disagree too much, i.e. the frequency difference is small, the attractor phase will entrain to the landmark pinning phase, but at a certain phase lag. And experimentally, that means that grid cells will phase shift when plotted in position in virtual space. However, if landmarks and path integration disagree too much, that means that two coupled oscillators have very different frequencies. They can't couple to each other and train each other. You get this complicated dynamics that at the level of grid cells would correspond to remapping. Okay? So basically, for small gain change, we expect a phase shift in the grid cells. For large gain change, we expect grid cells would remap. And I can't go through all the data slides and so forth, but we do actually see these two regimes. Just as a summary, here's example, two grid cells. Here's one grid cell that under a strong gain change, here's the activity before the altered virtual reality. Here's the activity after virtual reality is altered, and there's very little relationship between the grid cell firing pattern here and here. Here's under a weak gain change, you see there's just by eye, there's a phase shift. And of course, we did all the population summaries and all that and analyzed it carefully, and you can see all of this here. Reviewers, very tough reviewers, I would say unfair reviewers, made us perform new experiments at an intermediate gain change. They made us use our theory to predict which dynamical region we should observe at this intermediate gain change, and they made us verify our prediction. I think that's too much to ask for, but we did it. It all worked out, and you can read it in this paper. All right? Okay, so now let's just wild speculations, okay? What does Einstein have to do with what I talked about today? So, these ideas are out there in the literature. I'm by no means the first to suggest this, but there could be a very beautiful analogy between navigating physical space and navigating conceptual spaces. In fact, our ability to do higher order reasoning could have co-opted ancient evolutionary circuits to navigate physical space. In this analogy, updating position by integrating physical steps could be relating to updating deductions by integrating logical reasoning steps. And correcting positional errors using landmarks could be analogous to correcting deductive errors using experimental observations. And so, although we have a very, very long way to understanding neuroscientifically how we go from a Roomba-like intelligence to an Einstein-like intelligence, it's just absolutely awe-inspiring to think about how our ancient conceptualization of space dramatically evolved over the last 100 years to yield predictions about objects that are 53 million light years away, like a black hole. Thanks.
So, some questions. We have one here, another one there. So, please here first.
Hello. Yeah. Hi. You may have mentioned something related to my question, but I wanted to understand. So, the plasticity model, does it require the animal to experience the landmark? In other words, I was imagining I don't need to actually go to that door once I see all the landmarks in the environment for my brain to understand the map that I'm navigating.
Yeah, we were completely agnostic about how the landmark cell is triggered. For example, it could be triggered by distal cues. For example, visualizing or seeing a landmark a little bit far away at a certain distance. If that reliably triggers through perceptual circuits, a pattern of activity in the medial and entorhinal cortex, that's enough. You don't need to actually physically get there. In terms of just never moving around and just watching the room, that's a much more complicated problem, which this doesn't solve. That involves, I think, a lot of higher order reasoning that goes beyond just spatial exploration.
So your learning rule has kind of the counterintuitive, I'm over here. Oh, Surya.
I can't localize. Keep talking, I'll integrate.
Right here.
Okay, got it.
It has a counterintuitive thing of that you're calibrating the reality to the unreliable signal, right? You're moving.
Okay,
It's kind of, so I just wondered is there a disadvantage in that rather than using reality as the calibrator of the integrator?
Yeah, it totally depends, right? The reliability of a signal depends on context. For example, fixed locations in a high contrast environment could give you highly reliable information. But if you turn off the lights, then that becomes unreliable and path integration becomes more reliable. There's a parameter in our model that trades off the strength of landmark error correction against the strength of path integration in determining the position of the bump. And I suspect that the brain has higher order regulatory mechanisms to modulate that variable in different contexts, so that when landmarks are reliable, they have a strong impact. If landmarks, by the way, are moving around all the time, the plasticity rule will never learn strong synapses because there's no reliable correlation to pick up on. So at least that's solved automatically. But again, I suspect there's higher order regulatory mechanisms that modulate this gain parameter.
There's a landmark up there behind there.
Hi, Surya. Very nice work. I had a question which maybe at one level is a question of detail. I was curious about that result you showed that in a distorted box, the grid cell contours also got curved. But then they got curved paralleling one of the sides of the box and not the other, which seems like, it seems like it is exquisitely sensitive to which side of the box the rat first visits. Though, and there's this massive symmetry breaking that happens depending on which side of the box. But then that is a little bit counterintuitive because you also showed that the rat, in that one-dimensional track, the rat needs to go back and forth a number of times for things to for the elastic thing to settle and for everything to sort of become a smooth map. So it seems like an abrupt symmetry breaking from the first visit, and yet this smoothing which has to happen over multiple visits.
So a couple of things. You're absolutely right that there is a symmetry breaking process in this triangular environment as to which way the grid cells will bend. And sometimes, depending on initial conditions, it bends one way, sometimes it bends another. But you still need many rounds of exploration to settle down on one bend or the other. In a 1D environment, there's no symmetry breaking process. The couple dynamics of neurons and synapses is monostable. There's only one stable pattern. And you don't get bending, and you get the consistency condition solved. We're actually designing experiments with Lisa right now where we put put a mouse in a new environment where they can actually turn around and come back, so they can go back and forth. And we're going to record many, many grid cells at once. And then we're going to try to see on a trial-by-trial basis this consistency problem getting solved. So I'm kind of excited about that. So actually looking at the dynamics of learning in a new environment.
That's cool.
Perhaps I'm taking your cartoons too literally, but it seems to me that if a bump is associated with a landmark in box A, and now the animal is placed in box B, and the landmark is in a different position, there is going to be some sort of tug of war where the bump has to lose its association with the landmark that identified it in box A and switch to the landmark association in box B, and so there has to be some sort of winner-take-all competition process. How does the brain know which is the right one that it should associate the landmark with? Do you think that there has to be some higher level consistency between all possible bump positions and all possible landmarks in a new environment in order to select each individual landmark to associate with a particular position bump? And if so, who's looking at that higher order consistency?
There's a couple of answers to that. The triggering of a landmark cell could be contextually dependent. So there may not be a cell in environment A that responds to landmark A in environment A, and another corresponding cell, that same cell may not respond to landmark A in environment B. That's a possibility. Another possibility is that if there is some overlap, that could trigger remapping in complex ways that we don't fully understand. So that's another very interesting set of directions to go, is to make predictions about remapping when there's some ambiguity in landmarks. That's another very interesting direction.
Surya, the last slide where you propose these mechanisms for concepts, this paper by Tim Behrens, where he had a grid-like architecture for concepts, could you try to model something like that, or is it related to?
I mean, one thing that I like, for example, David Tank has some beautiful experiments where a mouse navigates an auditory space. It moves a joystick left or right to make a frequency go up or down. Is the person who did those experiments here?
Yes.
Okay, very nice experiments. You get place cells in frequency space, right? So that's,
Exactly.
Yeah, it's an example of co-opting spatial navigation for conceptual navigation.
Fantastic. Thanks a lot, Surya.